In this video we will discuss about a approach to enhance transfection efficiencies of these T cells. It is enhance transfection efficiencies of T cells using lipid nanoparticles. So why this research is focused on T cells? Because T cells are used in many immunotherapies such as CAR T cells immunotherapies to treat cancer cells. Although they are not that efficient against the solid tumors, but they can be used against the blood cancers with very much efficiencies. But there is a problem during the approach is that the transfection efficiencies of T cells. If researchers can improve these transfection efficiencies of T cells, then the amount of T cells required from the patients will be reduced significantly. So the idea is that the researchers, what they did is they took out the primary human CD4 plus T cells, and then they check this mRNA expression and activity in the using their different lipid nanoparticles. And what different lipid nanoparticles they used is that from the literature they are able to find out the components that enhance the transfection efficiencies of the lipid nanoparticle approach. That is phospholipid, lipid, ionizable lipid, and cholesterol. So they used the different combinations of these four components to find out the optimum compositions that will give enhance the T cells transfections, and they will validate those T cells transfections using mRNA expression and activity. So this figure on the left hand side is the abstract figure. The figure on the right hand side is the first figure. So it is similar to the abstract. That is, they took out the CD4 plus T cells, or they took out the T cells, then isolate CD4 plus T cells from the patient, and then they use these they design the experiment using these four different component of these lipid nanoparticles. And then what they did is they evaluate the single cell analysis. They did the single cell analysis to check the uptake and expressions of the mRNA. As in this case, they might have used a reporter in the case of this lipid nanoparticles encapsulated mRNA, and then they check these expressions using the flow cytometer. And in order to check the activity of this mRNA, they also investigate whether this mRNAs can be activated in the presence of tumors and can act against the tumors, and that we will discuss when we go through the results that are presented in their main figures. And after that, they also did this identifications of the T cells populations with this highest mRNA LNP response. And this highest mRNA LNP response means that they not only uptake high lipid nanoparticles, but they are also able to translate them to express those mRNAs on the T cell surface, as most of this mRNAs belongs to this CAR, and CAR is designed to express on the T surface or express on the cell surface, so that they can be used as a antigen to detect the cancers, to detect the cancer cells or the unhealthy cells. And then once they detect those unhealthy cells, they can trigger the activations of these T cells. At the same time, these T cells might also, if they are very modified, then they can also release some cytokines to kill those T cells. Usually T cells does not have this ability to release cytokines. The NK cells has the ability to release cytokines. So these T cells usually kills by triggering apoptosis within the cancer cells, and whereas the N22 cells or the NK cells trigger the apoptosis through two different ways. First, using the dead cell receptors, interactions with the dead cell receptors, or second one is by releasing the cytokines such as perforin and granzyme. But in the case of T cells, if they are not modified, then they will they will trigger apoptosis without releasing the cytokines. So I hope you understood uh, what this paper will be about, and those results we will discuss in the upcoming videos. That is to find out a optimum lipid nanoparticles compositions that can give us enhanced transfection efficiencies of the T cells. So that's all for this video. If you need to understand more about this paper, abstract and results in detail, results that although we have not discussed, you can go through the results in detail. You can go through the reference that has been provided in the description. Thank you for your time.